differences between complement receptor 1, interleukin 8, the receptor for interleukin 8, nitric oxide synthesis 2, and thrombin 4. So the genetic differences were observed between these patients in, for instance, complement receptor 1. Complement receptor 1 is actually a gene which presents pathogens to phagocytic cells. And this gene can be divided into 4 regions called A, B, C, and D. The SNPs we tested were present in region D, and region D is the binding region for MBL and complement factor 1. Interestingly, these polymorphisms we tested are also present in the McCoy and Sweng-Langley blood group antigens. So if we did find some SNPs in the binding region, theoretically this could mean that complement receptor 1 is less able to bind to the complement deposited on the fungal cell. We also found genetic differences in interleukin 8, the receptor for interleukin 8, and thrombin 4. And interestingly, for all those genes, the genotype correlated with high interleukin 8 production. So we wondered if interleukin 8 was actually present at the site of infection. So we too did some immunohistochemistry. And you can see here that interleukin 8 was also deposited on the fungal hyphae present in the grain and in cells surrounding the grain itself. So interleukin 8 was present at the site of infection. We then wondered if this was also found in the serum. So we took serum from patients and from healthy controls. And you can see that we found very high levels of interleukin 8 in our patient population in comparison to the healthy controls. These levels were actually much higher as reported in other infectious diseases. And the last polymorphism we found different was a NOS2 gene, which and the genotype we found correlated with low nitric oxide production. We found this also associated with lesion size, and interestingly, in the massive lesions we found also a phenotype associated. We found this genotype associated with higher nitric oxide production, which was comparable to our healthy controls. So we first looked if nitric oxide synthesis was present at the site of infection. As you can see in these pictures, it actually was. We then measured it into the serum. Nitric oxide itself you cannot measure into serum. You have to measure nitrite and nitrate. And we did find that patients had lower levels of these nitric oxide than healthy controls. We didn't find any differences when we divided the patients into patients with massive lesions compared to patients with small lesions. So in conclusion, we found some genetic differences between patients and healthy controls in the genes complement receptor 1, interleukin 8, the receptor for interleukin 8, thrombin 4, and nitric oxide synthesis. And the genotype correlated with lower nitric oxygen production and high interleukin 8 production, which was also demonstrated phenotypically. So this led to the following model. If this is the normal skin, then when the fungus becomes introduced, the estradiol level in the skin probably is at normal level. But when the fungus gets introduced, the level might rise. The fungus tries to defend itself and organizes itself in a grain. Neutrophils are attracted to the site of infection by the secretion of interleukin 8 from macrophages in the healthy tissue. The fungus then defends itself by producing melanin, because oxygen radicals are bound by melanin. And then the neutrophils will try to destroy the grain by secretion of nitric oxide. But since they have a defect in the gene, this production is lower than in normal people. So therefore maybe it's not able to destroy the fungus. So this was my presentation. if there are any questions.
Okay, so you're eager to actually answer some questions, but we can have questions from both speakers. But since Wendy's so interested in having some questions, maybe we could start off with a question for Wendy after that very nice talk.
So that was a really neat bit of detective work, but you showed that there was not very much difference in when you were looking at nitrate and nitrite in the serum between infected patients and non-infected patients.
Yeah, but it was statistically different.
But you would expect that the organism would have its own nitric oxide detoxification process. So if you've got heavy levels of infection, the organism may be detoxifying it, so you may not see high levels of nitric oxide derivatives in the blood anyway. So it'll be interesting when you've got the genome sequence to see what kind of detoxification systems they actually have.
Yeah, I actually know that the fungus can destroy hydric oxygen, like for instance Staphylococci can. So I know they have some defense mechanisms, but well, the lesions tend to be localized. So, and it's a correlation between what I found in the DNA and in the serum. But it would be nice, because this is only the beginning in understanding the pathology, to know something more about the pathology in mycetoma.
There may be other mechanisms. The nitric oxide goes through a peroxynitrite step before it's degraded, so you may find that there's other mechanisms to specifically detoxify peroxynitrite as well, so it's worth looking into.
Yeah.
Okay. Professor Hassan.
Thank you, Wendy. I think you made things, clear for me. I think, what you showed explains the pathology that I see. And I just want to ask whether you looked at toll-like receptors. Because, toll-like receptors are, the first line of defense, if you like, that they recognize certain, patterns and pathogens, viruses, parasites, fungi, and so on. So did you look at them? And if you didn't, would you think this is a good idea?
Yeah, when I was reading about this, I would like to do toll-like receptor 2 and 4, because those are implicated in other fungal infections, for instance aspergillosis. But it's only the beginning, so we can look at more genes, but also use animal models to study the immunology more clearly, because it's a field which is still underexplored, I think.
Okay. Professor Ali.
I just want to ask about pigments for both Wendy and Professor Ahmed, that fungi and probably other organisms like Pseudomonas, for example, they produce pigments, and Pseudomonas produces melanin. Now we have melanin here, and there is a hint from what Wendy said that melanin is probably a reducing agent that neutralizes reactive oxygen species, so it makes the organism more resistant. Does this apply, for example? I mean, I thought the melanin and the pigments produced by the organisms make them more resistant to our defense mechanisms and probably to our treatment.
In the case of Madrella mycetomatis, I demonstrated in the past that Madrella mycetomatis forms its melanin via the DHN melanin pathway, which is specific for fungi. And when you add this type of melanin to your cell culture media, and you do an in vitro susceptibility assay, interestingly, ketoconazole and itraconazole bind to the melanin, and the MIC rises 5 dilution times. So it becomes the level becomes as high that you cannot reach that level into serum. So especially ketoconazole and itraconazole are interfered with this melanin. Surprisingly, voriconazole doesn't show this effect at all. So indeed, melanin is very important in the treatment of mycetoma and also probably in the pathogenesis of it.
Okay.
And the other room as well, please.
Thank you for the speakers. My question probably to both Professor Hassan and Wendy. Just from the limited experience I had with the CNS mycetoma, I could say that the dura mater forms some sort of anatomic barrier to the spread of the disease. You know about mycetoma, it spreads largely through subcutaneous and subfacial planes. But it really finds difficult to penetrate the dura. I'm happy about that, but I just want to know how that happens.
I don't
Very
know.
wise.
But this is true. The case, that you're going to present, and we looked at it, histologically, it was just a small cavity full of grains and a fibrous, capsule around it. Whether, of course, the immune responses in the brain are different. It is a silent area. Perhaps the capsule occurs because the dura, reacts antigenically and forms fibrous tissue. So looking at, cytokines involved, in fibrosis will be quite interesting to find if there are differences between peripheral, tissue reaction and the changes in the. When you treat the patients with ketoconazole, you tend to have more fibrosis, and you again have a cavity, big cavity full of grains and just fibrous tissue around it. So, in short, I really don't know what is happening.
Usam Sharif, pathologist at Al-Gadarif. My question is to Professor Ahmed Mohammed Al-Hassan. Actually, sometimes we are asked to differentiate between bacterial and fungal mycetoma. And the only facility sometimes is H&E histopathological section. So we sometimes really find some difficulties in this. So thank you very much.
I think if I am working in a new area for the first time and looking at the histology, I would like all the organisms that are isolated to be identified precisely, by, and then looking at so many lesions, you learn that this is the commonest organism. And I report it as, for example, Madurella mycetomatis or somaliensis, depending on past experience and identification with, different methodologies, culture, PCR, whatever. Because, Sometimes even, here, you get other black grain mycetomas that are not really, Madurella mycetomatis, but other, types of organisms. And then, you will see the morphology is a bit different. And then you have to say, this looks rather different. Please do other tests to identify, the organism.
Could I take Chairman's privilege just to say how happy I am to hear you say that, because the organisms are clearly important and you really need to know what they are. And the point I think Wendy would make, and some of my other colleagues and some of our colleagues from here in Khartoum, is that to identify the organisms now and to find out whether or not they belong to the new species is very much easier than it used to be. And I would like to emphasize that it's not very expensive to do it. Of course, it's expensive to do it if you don't have any money at all, but if you have a little bit of money, it's not so expensive. So I would like to encourage people to do just what Professor Hassan said. I introduced him maybe as being the first man or the third man. I now think he's the first man because I really agree with what you say. But could I also ask a question? I did have a question about melanin. And the previous question, but one was talking about the comparison between the fungal infections and the actinomycete infections. Unless I don't think I'm wrong, but I think of all of the streptomyces pathogens, and I'm thinking more than just the somaliensis and probably more than sudanensis, none of them seem to produce melanin. So I don't know whether they produce melanin in vivo, but not in vitro. And Professor Hassan uses a term that I often use. I often refer to melanin and melanin-like pigments, but I don't actually know the difference. Do you? Between melanin and melanin-like pigments.
It depends on how the melanin, which pathway, which he refers to. And the first paper actually on melanin production was from South Africa, and they mentioned this, melanin-like, and I was not absolutely certain, really whether it is melanin or melanin-like, and you find it, in other, seeds. People here, they refer to these as communaswat. This is, اسمه شنو ها؟ نعم؟
No,
Yeah.
it's sativa, this is nigra, sativa nigra, which is this sort of a little bit hot spice.
Yeah.
You know, it's very small black granules.
Yeah.
Pepper. Black pepper.
It's not black pepper.
Black seed.
Black seed. I don't know what that. Anyway. Huh? Cumin. Black cumin. My daughter is more clever. And the patients actually describe it, as discharging these, black granules. They call it, communaswat. And the pigment, I'm sure, there is the same as in.
Well, in fact,
if I would make just a very small, perhaps a question, did you look at the changes in the blood? Because this is a very localized but very big thing. Does it make any changes in the differential white cells, in the level of interleukins and antibodies, and this? And could that be of any help? Because it seems it's a systemic disease, because these patients are actually wasted and maybe they have some other problems. My comment is the question asked by Sharif is really sometimes very difficult to differentiate histologically, but taking very good history, I mean, if they give the clinical and what they are getting out, and then if we look at it when macroscopically, when you see the black grains, or they are red or they are yellow, that's of great help, of course. It will put you in perspective. But then when you look at them, sometimes quite difficult really. Would you like to comment on the blood change?
Well, we did measure interleukin 8, we did measure nitric oxide synthesis, and we could measure that into the blood, because interleukin 8 was actually very highly present in the serum of the patients. So although it's localized, the levels are also measured systemically.
My first comment is to Dr. Sharaf. I think If you have excellent H&E, you will not need special stains to diagnose and differentiate the mycetoma agents. And there are good papers in the literature about this. And so I think what we need is a good technician to do a good H&E stain. And in fact, I put photographs in stack laboratories. If you have a look with H&E, you can see the differences between the different organisms. Because in the peripheral laboratories, you may not find facilities to do special stains. There was, back to Wendy, there were different views. At the beginning, some people thought that with pregnancy, the mycetoma improves. And Fahal showed you that it deteriorates. So I wonder whether with Wendy's studies now, not terminating pregnancy, but it seemed that it will have an effect on the prognosis for the management of the cases. And then we'll be in the problem whether to give ketoconazole or not to give ketoconazole. I hope if you have some views on this.
Well, of course, pregnancy is also hormonal, but it's not only hormonal. Also, the immune system is different in a pregnant woman as in a non-pregnant woman. So it's not an easy question to answer. I think you need to do a study on something about that. But ketoconazole can also be tricky, I think, in a pregnant woman, because ketoconazole itself has also some influences on the hormonal balance in men and women. There are some studies from which we've known that, for instance, progesterone levels are low when you're on ketoconazole. Testosterone is low when you're on ketoconazole. So I think you should base your therapy on previous experience. I presume you already have experience with ketoconazole therapy in pregnant women, and based on these experience you could decide if you want to continue ketoconazole or if you want to postpone it to after the baby is born.
This room 2, no questions from room 2. We haven't got any question.
Okay.
Have they slept?
Osama Ahmed Hassan, epidemiologist. I just want to comment on the prevalence of mycetoma itself among women compared to men. And in many publications we highlighted that the prevalence in men actually higher than women, like 5 to 6 times. So based on the field experience, I think the prevalence of mycetoma itself among women is underestimated. And this is actually due to many factors, like socio-economic factors. Many of the publication data, such as Sudan, for example, the context of Sudan, women have no good access to the medical service. They are not economically independent, so they depend on their husband like that. So, and the other issue is social stigma. So most of the women rendering are hiding from the clinics and health services. So what I would like to say that there might be a difference, of course, between male and female, but this higher prevalence in male, like 5 times, 6 times, I think it is questionable based on the field experience. Thank you.
I think this is a genuine difference. At the beginning, people gave reasons like this, that women don't go out, women don't work in farming, and Ahmed Fahal showed you how do women work in farming and how much they are exposed as much. And I'm pleased this ratio now, it's in Sudan, that's what we report, and we've seen many cases, Mexico, reported early on, it's 5-ton by Gonzalez Ochoa, now Benegol, and it is a genuine difference all through, and it's always higher. People may debate, is it 3-1 or 5-1? I'm inclined to believe it is 5-1. Thank you.
Over there.
My question is to Sandy. And first, I would like to thank you because you really took us beyond the normal in mycetoma research through your outstanding research. So after your findings regarding the gene differences, especially with the complement and interleukins, do you think the gene therapy would be a promising solution for mycetoma one day?
Well, gene therapy is not possible in any disease at the moment, but you could think of when there's more known about the immunology of mycetoma, so how it works, you could also try by supplementing, for instance, interleukin 8 or something like that, or any factor you find. But really we should know first more about the pathology than...
In the future.
Yeah, maybe in the future some immunotherapy is possible, but genetic therapy, that will take a very long time, I think.
Thanks.
Thank you for this elegant talk. Yusuf Allah, microbiologist in Sudan University. What do I expect to see if I crush this grain under the microscope on a slide? Does it contain conidia or mycelium or both of them, or this is one. Question number two, have you ever read about if the modes of transmission? We know the swans and the cats and dogs, tomorrow maybe. Is there any role for the insects for transmitting these diseases?
Well, the first question I think is better answered by you. But on the second question I can say that it's presumed that the thorn is important, but there's not really evidence that it's only the thorn. So termites could be a possibility as well. We found in our lab some evidence that probably patients have a Th2 predisposition, but we still have to prove that, and we want to use an animal model for that. But
insects like mosquitoes and bugs.
Yeah, there's no evidence for that yet, but that's simply, I think, because it's not studied yet.
I never crushed any.
I crushed some grains of a mouse model, and usually you tend to see some hyphal fragments with, in cement material. Conidia, I don't see them in grains because I never saw a conidia of... I can't get Margella mycetomatus to sporulate in my lab. I know there has been some studies describing sporulation in Margella mycetomatus, but it's not consistent. So people describe this type of conidias, others describe another type of conidia, but the definition of Margella in the Encyclopedia of Professor de Hoog is still that Margella is supposed to be a sterile fungus.
Can I respond to this? If you crush grains of Margella mycetomatus in potassium hydroxide, you see hyphae, and you may see chlamydospores within the hyphae. Now, the question of sporulation, I hope Sarah is here, and I don't know, Ahmed, is she presenting? Sarah Abdullah will be presenting, and she has managed to make them sporulate. But on the specimens, the clinical specimens, you only see hyphae and chlamydospores. That's all. Thank you.
Yeah, I have a question. And infections in general, it's related to exposure and how heavy the exposure and susceptibility of the host, immune, genetic, whatever. So have we investigated exposure in terms of inoculum size? How much of this thorn has of spores of Madurella or others? And that may have related to subsequent pathology or disease.
Well, in our animal model, we did different inocula, and we noted that we need a relatively high inocula of fungal mycelia to induce mycetoma. I don't think it's ever been studied on the thorns, because the thorn itself, if it is even present in the lesion, you cannot say how much was present at first place, but you don't see the thorn that often, I think, in the lesion, but maybe Professor Verhaal knows that better than I do.
Right. I mean, if we are saying, well, 1,000, they get exposed to the same conditions, but we don't know if their feet, for example, they have the same inoculum that get into the foot and then cause the infection. So it's not only susceptibility of the host and all the genetic, which are very nice and definitely a possibility, but also maybe the inoculum size, which can be very variable, depends on the condition, the places, the people.
And also another important thing which I didn't mention in this presentation, but under influence of sex hormones, the skin of a woman is also different as the skin of a man. So also that could be important why men more often have mycetoma than women. And that's also an indirect effect of hormones.
But again, how much of spores are getting into the skin, I think is maybe is relevant here.
Okay, a question for both speakers and also for Professor Majoub. Professor Majoub mentioned that perhaps people who develop mycetoma have some degree of immunosuppression, and that sort of predisposes them for developing the disease. You demonstrated that there is a type 2 response in patients with mycetoma. So it's a bit of the chicken and the egg. Which comes first? First, immunosuppression, and then this response is a result of that, or is a patient completely normal, immunocompetent, but develops a Th2 response after having developed mycetoma? Could you speculate on that?
Yeah, we recently had the hypothesis that Th2 predisposition is needed. So now we are doing some experiments in our lab with an animal model in which we induce the animals into a Th2 response and then try to induce mycetoma, because that's what we do in our animal model. And we do the same by inducing a Th1 response, and we want to see if mycetoma then develops, because then we know if it's a predisposition or if it's a result of.
Can I add to this? Have you any case with HIV and then expected?
Yes.
No?
Yes.
No HIV patient?
Oh, yes.
That will be interesting.
I can respond to this. Once we had an intelligent student from Jazeera University, and he said while in school they came around and they tested him for MON2 and he was negative, and they gave him BCG. Then he developed his mycetoma, and when he came to us, we tested him, he was negative and he had his mycetoma. So the negative test was before. Ahmed, this was one of the cases in our study of the immunological status. Yeah. And now you mention AIDS. Yes, there is a patient with AIDS who had a mycetoma. He didn't come to the center, and we might not talk about him now. Thank you.
It will be quite interesting. Okay. I think it will be very interesting to go into the community and test people, and follow them up over a long period of time and see, if they respond, differently. Measure their cell-mediated immunity. We did this in leishmaniasis in 2 villages. We tested people their peripheral blood mononuclear cells against Leishmania antigen. And some of these people, these were normal people. They responded by IL-4. Others responded by IL-12. We followed this up. And those people who had IL-4, they developed visceral leishmaniasis. Those who had IL-12 developed subclinical infection. Unfortunately, in the follow-up, we had a lot of dropout. But this was a tendency. And I think we are going to, repeat this. And I have been, asking, that we really should do this and have really long follow-up.
Yes. Just behind Mr. Badruddin.
Thank you very much.
Sorry, IL-4, is associated with the Th2 type of response. IL-12 is by the Th1 rationale.
I'm asking Professor Ahmed, and that I think the disease understanding, it needs intensive pathology work. There are many areas of the pathology and pathogenesis of the disease as unraveled. And Professor Ahmed probably has stopped in the position of formation of granuloma. He didn't pass to the fistulas, and later on to the granulation tissue formation, tumor-like doming form of the lesions, and the draining fluid that comes later on. The healthy condition of the granuloma formation, this is healthy, that ends up with sequestration and calcium formation and the ending of the disease. But the formation of the fistulas and so on, and the granulation tissue tends to exacerbate the condition more and more. This he didn't talk about. I'm asking him, do you agree with me? Do you think that the formation of the dome shape is a coalescence of granulomas together, leading to the fistula formation and draining out? Secondly, in answer to the question of our friend about insects as possible vectors, it comes from the answering of the pathology work and pathogenesis as well. You see the rash in the hands and in the legs and buttocks and so on. We think this might be due to insect transmission, that the multiple granuloma formation that you see the picture of the disease, it might be due to mosquito mechanically transmitting the disease from one area to the other. And we are in the veterinary lab, we're making a project like this. We want to study it in Medani, in Shigidi area actually. And we want to use mosquito blankets if we can restrain or control the disease and find there is a position for mosquito transmission as well as isolation of the organism. Thank you very much.
I'm not very clear, as to, what you have been saying. But if I find an agent and this all the time is producing, the same lesion, it's more likely that, the changes that I described are due to that agent. I won't say it is due to a fly or, whatever. So I think the changes, that you see pathologically are genuinely related to the presence of the grains and the organisms. And we know that in inflammation, you sometimes get, granulation tissue. Granulation tissue is found. And this precedes the development of fibrous tissue because it matures into fibrous tissue. Why, would I invoke something else if I have, a causative agent that I can really identify? Okay.
Okay, maybe the last two chances. Yes, please.
Sarah Abdullah. Thank you, Wendy, for your fruitful presentation. My question is that you mean the genetic factors of the neutrophil in patients unable to clean the first structure of the fungus that enters the body. Until now, we don't know, is it a hyphae or is it conidia or other structure. In my culture, a lot of conidia produced by Madurella mycetomatis strain sequenced and confirmed. But this genetic make those people more susceptible to other fungal infections, like, for example, aspergillosis, cryptococcosis, or other fungal infection. Did you report combination of mycetoma and other fungal infection in your patients that have impaired in neutrophil function? Thank you.
Well,
the last question.
These are minor differences in genes. They modulate some parts of the immune system. In order to develop invasive pulmonary aspergillosis, for instance, you need a very high immunosuppression to develop that. I don't have any data of other fungal infections present in these patients, but I think it's a balance. So there have been reports on other polymorphisms, for instance, in Dectin-1, which is associated with candida infections, with aspergillosis infections. So some of these markers probably are the same, but that doesn't mean that the patient always is prone to develop multiple fungal infections, because of course you also have the chance of becoming infected.
I would like just to emphasize that, anything in pathology is not just, a one-man show. It is a teamwork, and really should make, a group of people with different specialties, a pathologist, an immunologist, a mycologist, a clinician, epidemiologist. You work as a team, and that is what will get us ahead.
Okay, the last question or comment.
By amplifying these effective cellular immune response mechanisms?
Yeah. What is happening? For example, if in Madurella mycetomatis in particular, we did not look at, Th1 cytokines. We looked only at somaliensis. It may be different, I don't know. But anyway, what seems to me is that it is almost impossible to see a lesion that has been, completely, resolved. These type 1, 2, and 3 reactions, and the end stage of a granuloma. Is this end stage of granuloma, is it due to conversion from a Th2 to a Th1 type of response or not? That we don't know. And furthermore, even if I can demonstrate gamma interferon in the lesion, I will not be surprised that still the disease goes on. Why? Because we know that, for example, I had some patients that I have been investigating recently. Patients this time with leishmaniasis, and these patients were diabetics, type 1 and type 2. And I looked at, gamma interferon and other Th1 type of response. They were there. But these patients had very high levels of parasites in their tissues. Diffuse, leishmaniasis in these diabetics. And the answer here is that it's known that hyperglycemia in itself leads to immunosuppression, even in the presence of gamma interferon. Probably this is due to glycosylation of the end products of the interferon gamma and other cytokines. Anyway, so as I mentioned, we really should look into at the population level to early lesions and see if we remove this, we see evidence of complete destruction and change of the lesion into a granuloma with some remnants of the pathogen, but no active, hyphae. Then in that case, one can say that cell-mediated immunity plays a role, at least in early cases. In late cases, there may be antigenic paralysis because, there is a lot of, antigen in the area. So there are so many variables. And you need, a good immunologist, not somebody like myself, who looks at things and then disappears. You want somebody who really looks into the nitty-gritty parts of the immune response. Thank you.
Okay, I must say that has been a very, very interesting and very active, very vivid section with the superb presentations and very intelligent and nice discussions and questions. And before leaving the platform, perhaps the organizers or the Professor Fahal would like to make some announcement. If not, then would you like to make the final comment? Would you like to make the final comment?
You've almost said exactly what I was going to say. And I was going to say that we were very happy with the lectures we heard, and I think given all of the questions, the two that gave the lecture should be very happy with themselves. Thank you.
Thank you very much. I would like to thank you very much for being here all the day and for your active participation in this workshop or the international conference. As you can see around the corner, there is the e-learning Mycetoma Center. There is a lot of cases there. You can see, you can interact with these cases. There are questions, there are quizzes, there are feedback from these questions. So I think it will be a very interesting exercise to go and see these patients and try to answer the questions and the quiz in that. And there is a program for the mycetoma as well, the computer program for the mycetoma patient management. I think this is a great effort done by young chaps from the School of Mathematical Sciences. And again, you can see the program, you can give your ideas, and you can add in the development of this program. Tomorrow morning, we're going to have, hopefully, interactive session from Mecca by the internet, by the teleconferencing. One of our colleagues, Dr. Abdullah, he cannot make it to come to the conference, but he's going to communicate with us through the teleconferencing facility in the university here. So I will let you know in the morning whether we're going to start here or to send the other building in the hospital there where there is a teleconferencing system there available. In the morning we'll let you know exactly, because still having some problem with the IP address between here and Al-Qura University in Mecca. Once again, I would like to thank you very much for your active participation, and we hope to see you tomorrow for the 3 days. Thank you very much.